you all are aware about the wheat rusts. Wheat rust, these are one of the important component when we talk about the wheat system. Wheat rust, which is in this particular crop, there are 3 different species of puccinia which attack and cause huge economical loss. As per the predictions, yellow rust that is affecting around 10 to 12 million hectare area in North India. North India means the parts of J&K, Jammu and Kashmir, Punjab, Haryana, foothills, as well as some part of the Uttar Pradesh. And black rust that affects around 7 to 8 million hectare area in peninsular and central India. This particular area that comprises of part of Karnataka, Kerala, where the hilly region is there. So basically, the inoculum of this particular yellow rust that survive in the hill regions in the off season, but so far we did not get any type of clues regarding where this particular rust fungus inoculum survive in the off season. Although at the world level, especially in the China, they confirmed that berberis is playing one of the crucial role in collecting or in gathering different type of inoculums in the off-season seasons, and that particular inoculum that lead to the different type of infection in the wheat. So, if we talk about the Vitro cycle, everyone is very much aware that in this particular fungus, there are 5 different types of stages, starting from puccinia spores, Aschia spores, Iridia spores, Basidia spores. You are aware of this. But one of the stage, that is this particular stage, that is Iridospore or Ulitiospore stage. That is one of the effective stage, which is responsible for causing infection in wheat crop. And rest of the stages, and one beautiful thing about this stage is, this particular stage is multiplying around 7 to 8 times by using a sexual mode of reproduction. And if we calculated the loss caused by, if there are 7 to 8 cycles of these iridospores in a particular season, then it can lead to the loss of around 3,500 crore rupees. This is really a big loss. As I already mentioned you about this, this is one of the reason that this particular iridospores, the more the multiplication of this particular spore, the more the loss to the wheat crop. The loss in terms of the yield, because when it attacks, the photosynthetic area of the leaf is reduced, and ultimately the system of plant is disrupted and lead to the deformation of greens, small shriveled greens. And even if there is a 100% infection, then there is no yield at all. So this is a map of India. As I already mentioned, J&K, Himachal Pradesh, Uttarakhand, and New Delhi. So this is all the researchers which started research from 1931, Dr. K.C. Mehta, after onwards to till date. All they have concluded that and confirmed that the inoculum of stripe rust that is exists around this Ropad area or Punjab area, foothold of Shivalik Hills, but still not confirmed, and this is really one of the research issues, where the inoculum of this particular survives in the off season. So this is one of the big issue for doing the research in coming time. Similarly, if we talk about the brown rust and the stem rust, then this particular region here, this is the region where the Palli Hills, and the researcher thought that there is a probability that the inoculum is surviving this particular region. But as we evidented from the last 10 years, the incidence of yellow rust and brown rust, they are causing huge loss as compared to stem rust. Stem rust is just restricted to this area, but brown rust that are coming and moving towards the these hills, and now we also occurred a huge loss of brown rust in this particular region. So from this whole story, I tell to only two things. First thing is the importance of brown rust, which is so far restricted to the central area, is now coming towards the hilly regions. And second thing is the yellow rust, as usual, it is also one of the big problem, and it is still a continued threat for the wheat production. From this slide, my second message is very much clear that we still need to identify and decipher the variability, either in terms of pathogenic variability and molecular variability, so that we can devise beautiful strategies for the management of yellow rust of wheat. As we all know that the wheat system, that is very good example of a gene for gene hypothesis. I think everyone knows about gene for gene hypothesis. So the best strategies for the management of this rust is we use or develop different type of resistance varieties which are incorporated with the particular variety in which a particular pathogen is not able to attack or make the cultivar susceptible. So we are using the pyramiding the genes combinations and also study the variability. So we match the variability of the microbes or puccinia species that exist in the nature, and simultaneously we also match it with the varieties, what type of varieties they are existing to the nature, and accordingly we try to defend the attack of this particular fungus in Indian conditions. And from last 10 years so far, we have to successfully combat this particular disease. One example of the 1996, when there is a huge cultivation of PVW343 variety. At that time, there is due to mutations or recombination, there is an emergence of a new race that defeated Y9 genes, and it leads to the devastating effect on the wheat cultivations, and around 60% area of the wheat is totally restricted. And then we devise and develop a variety which have that particular gene, and then we have to successfully combat this particular crop from this particular races. And from last 3 years, there is also evolution of a new type of races into the natures, but we have a lot of stock with the different type of resistant genes which are into the wheat varieties, and we try to making not recommended to cultivate a uniform crop, but we try to use a different type of varieties that are suitable for a particular region. As I already mentioned that regarding yellow rust, if we go back into 2008 to 2019, then we get information that the varieties, the infection of yellow rust that is firstly reported from Himachal Pradesh, Shivalik Hills. From this is survey from 2008 to 2019 clearly mentioned that the Shivalik region is one of the hot spot region, because the first report of occurrence of yellow rust is from this particular ballot, and there are different type of varieties. So this clearly mentioned that there is some variation in the pathotypes. That's why they are particularly reported from different type of wheat lines. Just like HS 240, 295, 420, 43, like this, Burbot like this of varieties. So if we talk about the dominated wheat rust systems, although there are around 52 races of brown rust reported from India, around 30 races of yellow rust that is so far reported and preserved in Flowered Shimla, and around 12 races of stem rust that are reported and conserved in the station of Flowered Shimla. But all the races in the nature are not in a dominant or in a predominant fashion. So out of these, the yellow color highlighted region, these are the two prominent yellow rust races which cover all the wheat cultivated area in India. Simultaneously, these are the brown, this brown color, this is brown rust area, and rest black is this and this. These are the two prominent races that attack most of the wheat varieties the farmers grow in their field. But recently, during the survey, the different samples of yellow rust they are collected and compared with the barley. Then one of the beautiful information that come from that study is that the rust that attack on the wheat is also cause infection in the barley or vice versa. So just like here, 6S0 and 7S0, these are two pathotypes. They can attack both wheat and barley. So this is also really a big alarm, because where the cultivation of wheat and barley both are grown simultaneously, there is a huge loss for the emergence of the new type of races or new type of combinations. And from this one, 11S68, this is also one of the new race that also coming into the nature. So from the last 10 years, as I already told you, that 46S119, that is identified in 1996, was replaced by 78S84 pathotypes. So here the law of dominance, the basic law of evolution that occurs. The more the dominant pathotypes, more lead to the huge losses to the wheat crop. And the changing frequency of changing of this pathotype is going year to year up and down. So it is very essential to go for survey and surveillance and keep track what are different type of pathogens. Is it that particular pathotype is variable or not? So we have to analyze in the variability of this particular type of pathotypes in natures and collaborate this finding along with the laboratory studies as well as the field studies. So two-way approach: first collecting the sample from the field, then analyzing, then perform the analysis of the particular inoculum or iridospores into the lab, what type of race is this one, and simultaneously also study the molecular studies, the pathotype that are we have to multiply on the host. Is it the same or different? Because in nature there are different type of combinations, crosses going on, because the rust that is multiply or show variability due to 2 or 3 different mechanisms like mutations, recombinations, because this particular fungus has both systems, sexual as well as asexual variation. So there are 5 different type of mechanisms which operates and lead to the variation in this particular fungus. So if we talk about a variation in fungi, then simultaneously 2 different approaches. One is testing isolates, the powder of yellow rust, brown rust or stem rust. We collect from the field and go and multiply into the susceptible host into the laboratory and try match to the reaction on the differential host. Differential hosts are hosts which contain the nils line, near acidogenic lines. They have only a single gene which is responsible or showing the distinct type of infection types. So we multiplied that one and we have to match that criteria with the earlier pathotypes. Is it the same type of a spectrum or not? If that particular system provides similar pattern, then we think or we prove that this is the same pathotype. If not, then they might be a different type of pathotypes. But always what we see is not right. So we have to rely on the different type of DNA based markers. At the genomic level, at least a single change of mutations, because mutation is due to a change in alteration in 1 or 2 nucleotides. So we can also go back and analyze, no, this particular type of race has some change with the earlier race on the basis of this type. Then we can compare the results of both laboratory as well as the molecular science and confirm that no, this is a new race into the nature, and that can cause loss in coming time when it can be dominated in the nature. So I think you know about the different type of infection types. When we test any isolate on a particular genotype or nil lines, near acidogenic lines, we got the infection type 0, flag not, flag not 1, 2, 2 plus, 3 plus. What does it mean? This is the criteria, because in the laboratory conditions, we get a lot of informations. When we put a inoculum onto the susceptible host, it gives us some type of flag information. There is no spore, but there is some initial have sensitive reaction type reaction, that is flags. So we have to be up to from here to here, we count it as a resistance or multi-resistance line. But we cross 2 and 2 plus, then we move towards the resistance line. And on the basis of this, we have to categorize this particular type of a pathotype or isolate is effective or lead to susceptible that particular cultivar or not. Simultaneously, as we know that in the, just I gave here an example of puccinia, but in general the rule is same. We use a specific markers, and especially in from last 30 to 40 years, the people are using SSR markers, simple sequence repeat markers, for distinguishing at least difference of six nucleotides difference based on their awareness, and try to distinguish between the different type of strains, just like this. Here, this, and this, these are the same type of isolate, but when they get this allele, different alleles, this and this, then there is might be a possibility that it is the alternate form. These are different type of pathotypes in nature. And after that, we can go using different type of bioinformatics tools and go for designing of a tray. And based upon this informations, we can distinguish, okay, this particular pathotype Na3, that is quite distinct from this one. And this is the FST value, which give information of divergence, how one pathotype that is different from the others. So this is the beauty of both the techniques, how better we can analyze a particular pathotype and its variant forms. So far this introductions, but if we, the real works on this variability in the rust fungi is started by this great man. This is E.C. Stockman. He studied rust on different type of a host, barley, oat, sickles, and then come to the conclusion that the pathogens population, especially rust, that is mutate with time and environment conditions in any type of stress. It might be biotic stress, abiotic stress, and moreover, in nature, variability exists by two different mechanisms, either mutation or hybridization. Hybridization combines here everything like recombination, parasexuality, and different type of mechanisms comes under this particular theme. Now, what is variant? According to him, the progenies which shows variations in characteristics from their parents, deviation from their parent, that is variant. But when we talk about physiological specialization, this means there is a difference on the basis of physiology of a particular isolated strain. Simple. Within the species of a pathogen, there exist certain individuals that are morphologically similar, looking similar, but there is difference within their physiology, biochemical characters, and pathogenicity, and based upon this, they can be distributed on different type of host. So from this particular theory, the concept of use of different hosts come into picture, and from that period, we are continuously using the differential host for analyzing any type of isolate for pathotype analysis, and it works very well in nature up to this time now. So similarly, physiological races, a group of population within a species which have ability of infecting a particular genotype and differ in morphology but have physical differences. Okay. Similarly, variability, the property of an organism to change its character from one generation to another generations. This is very important, variability, because iridospore, they are multiplying in a production about 7 to 8 times, because a particular crop of iridospores, it takes around 7 to 10 days to complete cycles, and again multiply, again nature, and then go into different sexual forms, that is till iridospore formation. But we have no idea how the flush of these iridospores that cause variations among each other by different combinations. But further, within species, just like Puccinia gramis, there is form of specialists, and based upon different type of we can go and devise and nomenclature the race onto a particular isolates by inoculating on different host. So I am not covering much about this phenotypic or pathogenic variability here, but I think in coming lectures or a lecture you have to get some information about things. From here onwards, I go the recent advances in terms of understanding the variability at the genomic level. So this is a principle, how we have to go and conduct a survey for a collection of a particular isolate. Here I give you the example of stem rust, but the opinion principle is same for the yellow rust and the brown rust. Always when you visit a field, you will see that a particular patch of around 1 meter mostly 1 meter, that is effective as a rust. So try to collect 2 to 3 leaves from that particular field, and like this is your rust, and then cut or then invert both the leaves inside. The iridospores, they have to be on the inner side, not on the outer side, and club them and put into the envelope. This is the best way to save or to protect your iridospores, and then send to any laboratory for the analysis. So in the laboratory, what we do? We received your samples or any samples, and that after that we use some type of a susceptible host. So far, Agra local is one of the cultivar which is susceptible to all type of races which are reported from India. And one is in China, Chinese spring is another one which is also susceptible to all type of races throughout the globe. These are 2 types of genotypes which are used for determining or for multiplying the inoculum of a rust. All the 3 rust, maybe it is yellow rust, brown rust, or black rust. So we multiply inoculum because inoculum is so less to be tested on different type of cultivars. So first we receive the inoculum, then multiply on that one, and there is some type of a chambers. So that because we know that rust, it is an airborne disease. The inoculum spread from air to air. So we make a barriers so that a particular isolate is distinguished from the another isolates. So there are different type of chambers. They are closed chambers, and after that we develop some type of a disease development on this certain host, and after that we use that particular inoculum and test on the different type of a differential host. And after these type of combinations, in this host we use a 3 line system: A line, B line, and maintenance line. So these are the 3 type of lines in which one line contains a differential host, another line contains your whatever your varieties you are or whatever your germplasm you are going to screen that one, and based upon that cultivars we notice our disease reactions, and ultimately we use this type of a system to identify, okay, this particular type of spore when included in X, Y, Z, and so it giving a different type of response like 2 plus, 1 plus, 1 plus, 1 plus, and we have a record of all the isolates, and we match that recording with the earlier records of the pathotypes, and then we designate, no, this is the same pathotype or this is a different pathotypes. But one important point is that when we go for survey, this information must be there so that we can track like date, location, species, and cultivar if known, and then rust plus severity as a unique ID code, whatever these things. This is very important because when we go to when we know that this particular variety is there, so this provides first information this particular type of, because there works the gene hypothesis, and accordingly we designated, no, if this particular variety is there, so there is a probability of this particular pathotypes because it this particular isolate match with this particular gene. This is now when we talk about the molecular genotyping, what we did in this case, when collect the sample from the field, and in field we mostly prefer to collect young growth, young iridospores. Cut this and take this all the powder into the apron of tube. First criteria. Then after that, go for the DNA isolations. I think DNA isolation is not a big job. There are a lot of protocols, kits to isolate a DNA from this particular powder, and then use the PCR, use the specific markers relevant to that particular fungus. You can use either conserved markers because in fungus system there are some conserved regions which are present in every type of a fungus or species, but there is a variation within this particular region, conserved regions, that mostly used to differentiate one isolate or strain from the others, like ITS, internal transcript special regions, like beta tubulin, translation elongation factors, actins. There are a lot of different types of conserved regions we use and try to confirm or try to compare by using the sequencing. These amplicons using specific markers, then go to the sequencer and sequencing, and then compare the sequencing ATGC, ATGCC of one isolate with the another isolate, and then check out for the variations. You know, at point number 75 there is A, but another in the other isolate is T. So there is a probability that that particular isolate strain at genome level is variable from the earlier one or later one. So this is very much fine with the conservatism, but when we talk about the SSR markers, because SSR markers are multi-allelic markers, it provides a number of bands. On the number of bands, we can differentiate one isolate with the another isolates. So for that one, we have to make the Excel sheet. We run the gel, and we get the different type of bands. 1, 2. There is a, for example, in strain X, we get 3 bands of size of 100, 200, 300. In the Y, we get the one additional band of 400. Another, we have got the 7 bands, or another, we got the 3 bands. So we compile all the information to the Excel sheet. Okay, this particular isolate is giving 3 different bands. Another means 4 different bands. Another is 5 different bands. Like this, we have to club all the informations, just like here. Just like here. There are 227, 227, 227, X1, 227, another one, 35, like this. This is an example. Okay, and based upon we get a overall idea ki on the basis of different type of bands ki how variants one isolate or pathotype from the another one. So this is information from one of our publications in a crop protection journal, where we try to gather the samples of a stem rust of wheat from the different zones of India. Here, from all the part of India, from south or wherever we are from MPs, we collected samples and then use a different type of set. This is set 0, set A, and set B. These are the well-known different sets we usually use to characterize a particular isolate which we gather from the field. We take the sample from here and multiply it from here. So this is the phenotype of characterizations, testing your isolate on a particular host, and we get and conclude it to the one type of a set ki what type of reaction of particular pathotypes is giving on different host. Simultaneously, this is the different type of pathotypes, and we multiply on the host and then derived a Verlon's formula, because we already know that this differential has a what are the genes SR7A, 8B, 9A, like this. So this is very well and very well documented ki this particular pathotype has this Verlon's formula, this one has this one, this one has this one, this one has this. Our duty is just to compare the isolate with this profile. Then after this, what are the samples we have to get? We derive ki what type of a frequency of what type of gene is there, what type of urban gene is there. So we get the SR7Phi is more highly frequent, followed by this one. Okay, so this is a phenotype variability, but we try to compare with the different type of SR markers. So in the literature, there are already different type of markers, functional markers, and we try to differentiate different type of a predominated pathotypes based on this one. So we get that 2142 and 151A and 11A. These are 3 different type of pathotypes. They are coming to one clusters. What it means? It means they have some phylogenetic relationships among this. Maybe there is an evolution of this particular phenotype is linked with this one, this one, this one. But the evolution of this particular pathotype or race is not similar with this one. They have different evolutions. So this clearly mentions that in nature there are different type of forces which act and creating evolutions. There is not a single strain which lead to this one, but there are different type of things which are going to the nature, and we need to understand this, all the variability mechanisms into the natures if we really want or decipher a particular type of a strain. Similarly, as I already mentioned that if we, beside SR marker, if we use a conserved markers like ITS, beta-toluenes and ketopentose reductase type of genes, we also get different type of clades. From this basic systems, we get the evolutionary history, ki how fitness our particular strain pathotype in nature is. So in this case, we justly used all the well-conserved and preserved pathotypes that exist in the Flowerdale and sequenced by using the markers that derived from the ITS, intertask aspersion, beta-toluenes and ketopentose reductase, and then go and characterize this one. From this information, it is clearly mentioned that the evolution is on the 2 different places. The PST-A and PST20A, they have to be evolved differently than compared to this one, because there are 2 clades. And even in the first class, there are 2 different types of a populations. One is this one, this one, this one. So the evolutions is quite distinct among the isolates or pathotypes we used or preserved into the Flowerdale Shimla. So similarly, this is a formula, everyone's formula, and different type of evaluation is coming here. Now, if we compare these styroformes, yellow rust, stem rust, and brown rust, what it indicate? It indicates that all the three races, they have to be evolved differently, not on the similarly. Not like that single pathogen is there and it lead to the another iridospores and it can cause to stem rust. No. So this clearly shows that the evolution and phylogenetic relationship of all the three races are distinct. Similarly, this is based on the beta tubulin regions. All the three are quite distinct with each other, either brown rust, stem rust, or the leaf rust. So far, but it is quite difficult to interpret the information based upon the single gene system like ITS, beta tubulins, because the races, they are multiplying at a fast rate into the nature. So the peoples, they are trying and compare these populations based upon the whole genome analysis. So far, if we compare the up to 2019, there are a lot of pathotypes, just like Puccinia titicina. This is brown rust, 77106. These are two races that are whole genome sequence from India, and this is from abroad, from Canada. Similarly, from Puccinia gramis titicii, this is from abroad. But in case of Puccinia styriformis, there are different type of races that are sequenced from India. At least 13 races that are sequenced, whole genome sequence from India. Then further, based on the whole genome analysis, the peoples trying to compare the different races. What is the mechanism of evolution? What is the mechanism of fitness? And how the things going when we analyze a particular isolate at the whole genome level. So based on these informations, they conclude that this particular pathotypes is quite near to this one, but quite distinct from this one, which is USA. This pathotype is from USA. What it mean? It clearly mentioned that the pathotype that exists in India is quite different than that is present in USA. This means the inoculum that comes from abroad or by wind is not reached here to cause infection to the Indian conditions. So this is the beauty of a genome information. We can replicate the data from one lab to another lab, and we get much more information. But this is the pathotype from China. This comes within this clade. What it means? It means the system of China is also different from that one and quite outward, because there are two clusters, one this one and another is this one. This means that the races system that exists in China and USA, they are quite different, at least in comparison to these pathotypes. Similarly, we also compared about the evolution of two different pathotypes in this particular research paper, where 31K and Y valences. One is reported from 1936, another is reported in 1982, and another is in 1996. To find out, to prove the hypothesis, is this particular pathotype is evolving again and again and leading to the generation of a new type of pathotypes, or is there some different mechanism that is operating into the nature? So, similarly in the system of bacteria system, there is a 16 SRA region. So the same feature of that particular region. This is a conserved region, but there is a variation within that particular regions. Effector protein. What is effector protein? Effector, effect, anything which produce any effect. The thing or any entity or any molecule secreted by pathogens which produces any typical symptom or certain reaction in the host is a effector gene. Fungus releases something, either some enzymes or different type of proteins or some type